The additional biology part 2 hangout. Again, hopefully you can all hear me okay. And if you've got some paper and a pen, along with a calculator, you'll get the most out of this session. So if we have a look at our previous session for the additional biology, we covered the following topics. We covered cells, organism distribution, plants, and enzymes. So for today's session, we're going to be looking at DNA, genetics, and life on Earth. So let's take a start. And again, as we go through, if you've got any particular questions and you're on frog, just post them and I'll do my very best to answer as we go through. So the very first part to this session is DNA and the structure within. So specifically for DNA, well, DNA is our instruction for life. It's the information which tells an organism what to become, how to grow, what features to have. And we find it wrapped up as long molecules. Now, these long molecules of DNA are found within chromosomes. And the chromosomes come in pairs. We have 23 pairs or 46 chromosomes in every human. And specifically, a small segment of DNA is known as a gene. And this goes to code for something. It normally codes for a particular protein. The final thing around the DNA and the DNA structure is where we find it. And we find it within the nucleus of our cells. So DNA is the information for a particular organism found within the chromosomes, the instructions to make an organism become what it is. And within that, they're all found within the nucleus. Now, the final point with our DNA is this. Everybody's DNA is unique. So no one's DNA is the same as anyone else. There is a slight exception to that. If you're an identical twin, then your DNA is in fact the same. But everyone else's DNA is individual. And so this can be used for DNA fingerprinting, for paternity testing to find out parents, any kind of forensic science. It becomes particularly useful. And they'll always be given at around about 99% accuracy. There's always this tiny, tiny, tiny chance that the DNA is not that of the suspect, because perhaps there is an identical twin. But generally speaking, we are incredibly precise when we come to look at DNA. Now, with our whole section on DNA, we need to look at the way that an organism replicates. And replication comes in two forms. It comes either via mitosis, which is what we'll look at to begin with, because this comes up for everybody in the exam. And it does come through another process called meiosis, which we'll come into in a second. So when we're looking at DNA replication, we might be drawn to something like this. And these are the 23 pairs of human chromosomes, of which there are 46 individual chromosomes. And you'll notice the 23rd pair is the XY. And we'll talk about that later on. But these essentially are our sex chromosomes. So what is mitosis? Well, mitosis is essentially asexual reproduction. It's essentially cloning of a particular cell. It's an exact copy. And it works via the following. Well, first of all, we start out with a completely normal parent cell, a skin cell, a gut cell, an eye cell, a normal cell. In fact, any cell that isn't a sex cell and isn't a gamete. And when we look at this normal cell, the DNA is completely spread out. Well, the very first point for mitosis is this part here. We get a DNA duplication. It doubles up. The chromosomes, each arm of a chromosome is a duplication of the other. And so we can see here in this particular part, we've got a doubling of our chromosome. This particular chromosome is doubled, and this one, and so on. Well, now we've got a doubling of our DNA. Essentially, we've got 200%. We get our third stage. This is where our chromosomes line up. And they line up, and they're then pulled apart from very specific ends. So we have these spindle fibers that pull them in half. Each half of the chromosome goes in one direction. And that leaves us with this. We've now got ourselves two nuclei of each cell. Each arm of the chromosome, which was a duplicate of the other, has found its way into one nucleus. And so these two are identical in their DNA. And eventually the process is complete when we get a cytoplasm dividing to leave us two identical cells. So for mitosis, we're having identical cells being formed from one original parent cell, and we're getting that by a single duplication, an even split forming to our two cells. So that's mitosis. That occurs in every cell of our body apart from our gametes, our sex cells. They do something slightly different. So let's take a look at the second type of cell replication. This is meiosis. Now, meiosis is all about producing gametes, the sex cells. We've got an egg and eventually a sperm that will fuse to one another in fertilization to end up with an embryo of 100% DNA. They both carry 50%. So meiosis is a way of getting half the DNA. One copy of each chromosome, no pairing. Well, let's take a look how it works. First of all, it starts with a normal cell, a normal body cell, 100% of the DNA was within. And just like mitosis, for the first part, we get a DNA duplication. It doubles up. However, the difference with meiosis is actually we get two divisions. Well, here's our first division. At the first division, we find ourselves, just like we had in mitosis, the chromosomes line up. And they line up in the very central part of our cell. Now, at this point, the pairs are pulled apart. So each new cell only gets one copy of the chromosome. So it might be, for this example here, if we take a look, during our part, at the second division, we're finding that the chromosomes again line up and again are pulled apart during our second division. But there's a slight subtlety here. They haven't lined up perfectly down the central line where they are pulled each chromosome half in half. What we're actually getting is a slight randomization. So for example, in this cell here, for example, if the red chromosomes were the mums, we're finding that we've got 3 of mum's chromosomes, 3 red ones, to 1 of dad's. And vice versa over here, we've got 3 of dad's to 1 of mum's. And so when these are pulled apart, they're pulled apart in such a way that actually we end up with 4 gametes, each containing half the DNA, but most importantly, it's slightly randomized. In other words, this particular gamete has a certain amount from mum and 1 of dad, 3 from mum and 1 of dad. Whilst over here, these gametes have 3 from dad and 1 of mum. And conversely, they're different sides of the chromosome have been pulled between the 2 parts. In other words, we're getting these 4 gametes with a single set of chromosomes in that have initially come from 100% of one of the parents. They're down to 50 and they're random. And this is why, for gametes, we find siblings from the same biological mum and dad, for example, 2 brothers, can be quite different. They've got 50% from mum and 50% from dad, like one another. But one of the siblings might have got, for example, mum's blue hair, whilst the other brother might have got dad's green. And this comes because of 2 divisions, of which one is randomizing our chromosomes. So meiosis is giving us 50% DNA, unlike mitosis, which gives us 100%. Now, meiosis will only tend to come up in the higher paper. The key bit to remember is the randomization. Now, thirdly, for DNA, is this term stem cells. Well, stem cells, embryonic cells, haven't differentiated. They haven't become anything yet. They can turn into any cell. And this has hugely beneficial consequences. Essentially, we can potentially cure a huge number of illnesses. So if we take some of these stem cells from our embryo and grow them, we might have someone who's got perhaps a slightly dodgy heart. Some of their muscle cells within their heart are not firing correctly. Well, potentially, stem cells could be used to treat them. They could be grown into specific heart tissues. Or perhaps they could be grown into insulin producing cells in an individual that lacks it. Or perhaps for someone that's paralyzed themselves, they can be grown into new nerve tissue. So a stem cell can turn into any cell, offering a huge amount for medical research for curing diseases. But there are some negatives. and you need to be able to argue these in the exam. Specifically, we're taking these stem cells from embryos. And so people will argue that they shouldn't be used for any kind of medical research because potentially they're a human life and you're destroying them in the process. The counter to that is perhaps there's someone who is currently alive who is suffering. And by using stem cells, you're potentially taking away their pain. You've also got the argument that most of the embryonic cells come from IVF, cells that are over. And so they were going to be destroyed anyway. So you need to be able to argue the benefits as well as potential negatives. So they're the questions surrounding DNA. And as always, we're put on a couple of exam questions for you to have a look at. And the timer is in the top right for you to be going ahead of me. But again, I will be answering them relatively quickly. So you might want to pause at this point, try and answer, and then come back into it. So let's take a look at this particular question. The diagram shows one way that stem cells can be produced from human embryos. And we've got a fertilized egg, a 5-day-old embryo, inner cells of the embryo removed, stem cells, and these can become blood, nerve, or muscle. And stem cells can be used to treat conditions such as paralysis. For 2 marks, explain why. Well, essentially our 2 marks are going to come from saying stem cells can differentiate, can become any cell. And that cell might be something such as nerve or muscle, which might be the reason this particular individual is paralyzed. Perhaps they've got some muscle wastage, or they've broken their spine and they snapped their nerve. Well, these stem cells could become those cells. So the stem cells can become any cell. And for the second mark, just an example of one of those 2. Now for the second part, during pregnancy, the umbilical cord and the placenta join the embryo to the mother. At birth, the umbilical cord is cut. Stem cells can be obtained from the umbilical cord. Many people think that stem cells for treating human conditions should be obtained from umbilical cords rather than human embryos. Suggest one reason why. Well, essentially, this is any type of ethical argument. There's an ethical issue with destroying embryos. Potentially, that is a human life. Getting them from the umbilical cord means we don't need to do that. And finally, stem cells divide by mitosis. Gametes are formed by meiosis. Give 2 differences between mitosis and meiosis. Well, mitosis is 1 cell division. Meiosis is 2. Or mitosis gives us identical cells. Meiosis gives us half the DNA. Or mitosis gives us 2 sets of chromosomes. Whilst meiosis will only give us 1. Okay, the second question for DNA. We've got ourselves some cells in a root of an onion plant. we've got cell X and Y. Cells X and Y have just been produced by cell division. Name the type of cell division that produces cells X and Y. Well, this will be mitosis. We're in the root of an onion plant. Nothing to do with the sex cells of a plant. So it's mitosis. What happens to the genetic material before the cell divides? Well, this is our first doubling. We get twice the amount. Finally, a gardener wanted to produce a new variety of onion. Explain why sexual reproduction could produce a new variety. Well, when there's fertilization, we've got ourselves the fusion of gametes. In other words, we're getting genetic material from two sources. We're getting a mixing. We could say we're getting one gene set from each parent. We could say that meiosis leads to gametes, and gametes are varied. So any of the points that we've got gametes, we've got a mixing of genetic material, perhaps the gametes have only got half the DNA, one of the chromosome sets. So that's DNA. Now, the second part to the second half of biology is genetics. And we start with a chap called Mendel. Now, Mendel was a monk. He lived in the 1800s, and he was carrying out his work around the 1840s, 1860s. And he was essentially experimenting with peas. And he was looking at their features, what they looked like. And he ended up coming up with a variety of very important comments and very important theories. He essentially discovered the term hereditary, getting something from your parents. And he also came up with the terms dominant and recessive. We're going to need to look at how he did it. Well, it starts as follows. Mendel took himself a purebred, tall plant, and he crossed that with a very short pea plant. And he found out what happened. Well, when he crossed, and we'll talk about how we do this in a second, these two plants, we need to be able to use the Punnett Square method. So these two here are my tall genes of my tall plant. And these two here are my other parent plant, my short genes. And this is what Mendel found. When he crossed them, he got one tall gene from the first plant with a short gene from the second, and so on and so forth. In other words, in this first generation, all of the offspring were 100% tall. But he then did the following. He took this first generation and he crossed them. If you remember previously, they all were big T, little t. In the second generation, when he crossed his first, he then found the following. Big T, big T, big T, little t, big T, little t, little t, little t. He came to the conclusion of this 3:1 ratio. 1/4 of the F2, the second generation cross, were short. All the rest were tall. But actually, he went one stage further than that. If we look closely, this tall plant has 2 dominant alleles. It is homozygous dominant. Both the same. We've got our homozygous recessive. The short plant has 2 alleles, both the same, but they're recessive. It makes that particular plant short. And we then have 2 other tall plants. So we've got a total of 3 tall plants in total. But these 2 were big T, little t. They were heterozygous, but they were still dominant because the dominant allele was winning. In other words, Mendel came up with the idea of alleles. These, and we didn't know about genes at this point, but these different versions. So we could find that we have genes that make something tall or genes that make something short. And depending on how they mix from both parents, gives us our particular offspring. So we need to look at a few examples of this to make sure we can draw it in the exam. And I've been using a Punnett Square to do it. Let me give you a slightly easier example. Say you took a male and a female human and you wanted to find out the ratio of if you're going to have a boy or a girl. Well, hopefully most of us would be able to say that we've got a 50-50 chance. But how do we get to that? Well, if we take the female will have XX on her sex chromosomes. She will produce an X egg or an X egg, only ever X eggs. The male, on the other hand, will either produce an X sperm or a Y sperm. And hopefully you'll see how this comes about. So in this particular case, at the top here, I've drawn my female running along here. And down the side, running vertically, I've drawn the male. So when we cross them, I go down my female X, down my female X, and on this column, down the female X, down the female X, and there we go across. Here's the X sperm from the male, XX, and X goes across. I've got 2 females. But I now need to run along with my Y, XY, XY. I've got 2 males. So I have a 50-50 ratio, a 1 in 2 chance of it being a girl and a 1 in 2 chance of it being a boy. Now, along with genetics and Mendel, we need to look at some of the disorders. And again, we'll use the Punnett Square method to talk through them. So The first that we're going to look at of our genetic disorders is something called cystic fibrosis. Now, cystic fibrosis is essentially a disorder of the cell membranes. It causes a lot of mucus to build up. It ends up, unfortunately, essentially meaning you are struggling to breathe as fluids build up. It's particularly nasty. But luckily, it's on a recessive allele. Dominant alleles will always beat recessive alleles. So the classic one, someone with brown eyes might have the brown allele and the brown allele, but they could also have the brown allele and the blue allele. But the brown will always win. Someone with blue eyes will only have the blue allele. It's recessive. If the dominant brown ever came along, they would be given brown eyes. So to have blue eyes, you must have both recessive alleles. Well, cystic fibrosis is itself on the recessive. So it would be a small c. So let's take some examples. If you had two parents that were not sufferers of cystic fibrosis, but were carriers, they would look like this: big C, little c, big C, little c. We know that they are not sufferers from it because it must be on the recessive, and they've got a dominant allele each, so that one will always come through. But they are carrying it. That word carrying means that they have it, although they're not showing it. They have genotypically got it, but phenotypically they're not showing it. The converse to that would be a completely healthy human would have two big C's. A sufferer would have two small c's. So with that in mind, what are our chances of getting cystic fibrosis? Well, actually, this goes back to Mendel's original work. We've got two C's here. We go down and across. We've got a big C and a little c, but the big C will win. We've got a big C and a little c. So these three people are fine. One completely normal person and two carriers, but not sufferers of it. But unfortunately, this person will be a sufferer of it. It's our 3-to-1 ratio. Now, that doesn't mean if you have 4 children, the fourth one will get it. It's just a ratio. You could have 10 children and they're all absolutely fine. You could have one child who isn't. But statistically, if you had enough children, it would go to the order of 3-to-1. Now let's take a look at a second disorder. Let's take a look at something like polydactyly. This is extra digits. So there's someone with 5 fingers. Polydactyly is someone with an extra one. It's on the dominant. So anyone who doesn't have polydactyly will never have the dominant allele. You won't ever carry polydactyly. You'll either be someone that doesn't have the disorder or someone that does. So let's look at this. Here is a particular individual with polydactyly, and they marry someone and have some babies with someone who does not have it at all. What do we find? Well, big D, little d, big D, little d. These two will have it. Little d, little d, little d, little d. These two won't. We're at a 50-50. That being said, if both people had polydactyly, even if they've only got one allele with it, we would find that we get big D, big D, little d, big D, little d, little d, little d. We're 3 to 1, but in favour of getting it. Furthermore, as soon as you start putting in someone that has both alleles carrying it, and perhaps someone that is simply or is still suffering but doesn't have both alleles carrying, we suddenly find ourselves in a position where every single person will get polydactyly, 2 of them for both alleles. So you need to be able to draw Punnett squares. The rules are parent 1 goes along at the top in part 1 and part 2. Parent 2 goes down the side, and you simply cross them up, down and across, down and across, down and across, down and across. There's no right or wrong, but rules of a convention, the capital letter being the dominant normally goes first, followed by the recessive second. So these are Punnett squares. Looking at a few genetic disorders, when we talk about all of our genetic disorders, it's then prudent to talk about embryo screening. This is looking at our embryos before the baby is born at a very early stage. It might be the case that perhaps you have people in your family who suffer from cystic fibrosis, and you want to find out the likelihood of another baby having it at a very early stage of pregnancy. Well, the positives for this are perhaps you're reducing the suffering of someone. Perhaps cynically, you could say you're reducing the costs involved in allowing someone to live with this particular disease over many years. Of course, the flip side to that, embryo screening, although it might show you this disease, it doesn't necessarily mean that life is anything less important. Although there's a monetary cost to it, that's all it simply is. Screening that embryo will mean potentially the parents decide to destroy it. And there's arguments against life there. So you need to be able to argue for both ways in the exam. Yes, of course, there are some positive implications of screening these embryos, but that is counter to, essentially, the destruction of life. Now, I've just had a couple of questions before we move on to the disorders and a few other examples. Specifically, a female produces 1 egg. So how do we get the 2 chromosomes? Let's not forget, there's 1 chromosome from the female. It's always going to be an X. But don't forget that egg has half the DNA in it. The other half will come from the sperm, and that sperm can either be an X sperm or a Y sperm. Depending on which one meets up, if it was this top one, we'd get XX, we'd have a girl, and if it was this bottom one, it would be XY, and we'd have a boy. So hopefully that's answered that particular question. Okay, our final part for diagrams, I'm going to show 2 examples here. The first are on the left. Now, these are people that might be carrying something or might be suffering from something. Again, in the exam, they might draw them like this, but it's just like doing our Punnett square. So I've got parent 1 here who is big F, little f. So they are heterozygous dominant. They are normal, but carrying it. And in fact, this is the same for both of them. So if we were going to draw our Punnett square, don't forget I've put one parent along the top and one parent down the side. We would get ourselves big F, big F, big F, little f, big F, little f, and little f, little f. This one is here, is here, and these two are here. So a Punnett square is a very simple way of working out the ratios of what we're going to get. Again, in this case, again, a 3-to-1 with 1 being our sufferer. Now, typical exam questions point towards things like this right-hand diagram. They give you some kind of key. I've got two people. I've got John and Susan. John is a male, and he's neither a carrier nor the sufferer. So he will have himself two dominant alleles. He will be homozygous dominant. Whilst Susan, the circle will denote she's a female, and she is a carrier. It's half filled out. The question might come to something like, with this couple, what's my new baby going to be like in terms of our ratio, our percentages? Well, we've got Eve, a female, and Phil, a male, but they're both carriers. So when we come to draw our Punnett square, we would be looking at a carrier that isn't necessarily a sufferer. So this is going to be something on the dominant. If it was on the recessive, we would find that in this particular case, you wouldn't be able to carry it. You'd either be absolutely fine or you'd have the issue. So let's take a look with our new baby here. We've got ourselves Eve, who's carrying it, and we've got ourselves Phil, who's carrying it. Neither suffering, both carrying. Well, our ratio would go as follows. 2 big C's, 25%, absolutely fine, so neither carrier nor sufferer. 25 and 25, big C, little c, big C, little c. In other words, 50% carriers. This leaves us little c, 25% sufferer. But it's our 3-to-1 ratio. Technically speaking, we've got these three, this 75%, absolutely fine. They are carriers, but phenotypically, they're not suffering at all. Whilst we've got 25% here, but are suffering. Okay, let's take a look at some questions. Some babies are born with extra fingers or toes, as shown in the photograph. And this was, as we previously called it, polydactyly. Diagram shows the inheritance of polydactyly in a family. We've got our male and our male, our female and our female, and some are unaffected and some are affected. For the question, polydactyly is caused by the dominant, capital D. The recessive allele of this gene is represented by little d. Use one genetic diagram to show the inheritance of polydactyly gene by R and S. Well, the first thing to look at is R and S are coming from the parents, P, which is a male that is affected, and Q, which is a female who's fine. So if we were going to draw our male that is affected, we're going to draw the following. We're going to draw big D, little d. I'll come on to why it's not big D in a second. That male will have babies R and S with Q, who was unaffected. And we know it's on the dominant, so for her to be unaffected must have two alleles that are both small. Excuse me. So what do we find? We get big D, little d, big D, little d, little d, little d, little d, little d. We have a 50% chance of R or S having the disorder and a 50% chance of them not. And this is in fact between between R and S what we found. It's a 50-50 for them. And that will get us our 4 marks. Now the reason we know it can't be big D for the affected male is if it was, and that affected male went with a female that was completely fine, wasn't affected at all, would get big D, little d, big D, little d, big D, little d, big D, little d. They would all suffer from polydactyly. And we can see quite clearly in P and Q's offspring that S, this particular one here, is an outsider, but O and this particular one aren't suffering. Specifically, of the offspring of P and Q, these two do not suffer. And so it couldn't possibly have been big D from the father. A subtle point that you need to start by looking with the diagram to begin. Now, the second part of the genetics question is a spelling, punctuation, grammar, a 6 marker. They're asking for the benefits of screening for embryos for genetic disorders. Many people might favour this for cystic fibrosis, but not for polydactyly. And we need to compare the issues, talking about both conditions as to why. Well, again, we might get some of the following. If we're talking about cystic fibrosis, we could be talking about reducing the number of people with it. It's a very serious health condition. Again, it will have cost implications. Screening it will give the parents some consideration about whether or not they should terminate the embryo. But of course, the negatives with all of this with cystic fibrosis is potentially if we're screening, we might cause damage to the embryo. And if we choose to keep the embryo and it's got damage because of a screening, that might have quite serious implications. There's a risk to mum. Any ethical considerations, really? Like we said earlier, the positives versus the negatives. The flip side to that is something like polydactyly. Well, this is simply, and I'll use the inverted commas here, a disfigurement. There's no real health implications. It's not life-threatening. So going through embryo screening to essentially change this can be quite unjustified. There's a great deal of risks to doing this. You can argue those risks because cystic fibrosis is a particularly nasty disorder. But polydactyly isn't. It's not life-threatening. And so there's an argument either way there. And again, with a 6 marker, be aware of the spelling, the punctuation, and the grammar. Now, for the second part, we're looking at Mr. and Mrs. Brown. They've got a child with cystic fibrosis. They hope to have another child, and they want to know the probability that their next child will have cystic fibrosis. They visit a genetic counsellor who explains, and here's our key word, you're both heterozygous for cystic fibrosis. There's a 1 in 4 that your child will have it. We've got N for being unaffected. We've got small n for having it. And then Mr. and Mrs. Brown have the same genotype. First question, what is their genotype? Well, it's big N, little n. We know that because the clue was in the paper. They are heterozygous. They must be different rather than homozygous. If they're both big N, little n, what are their chances of 1 in 4? Why? Well, big N, little n, big N, little n gives us big N, fine. Big N, little n, fine. Big N, little n, fine. Little n, little n. Potentially cystic fibrosis. 3 in 1 or a 1 in 4 chance, 25%. Okay, on to the final part. The final part is life on Earth. We're going into a little bit more of an addition from the core science where we're looking at fossilization, we're looking at evolution, and we're looking at speciation. So with all of this, remember, fossils are the remains of dead plants or animals from millions of years ago. And they've had a gradual replacement. The body tissues or the impressions, in other words, the footprint of something, have been replaced by minerals. They've mineralized. They've become rocks. Well, these fossils give us really good evidence for what life was like long ago. When things fossilize, the oldest things are found at the bottom, the newest at the top. So our dinosaurs are down here, our humans, if they were to fossilize, of course, would be much higher up. And so it gives us our evidence of what life was like. It shows us the change. Of course, that doesn't answer the original life on Earth. We think they were soft-bodied, but of course, being soft-bodied means that fossils didn't form. There's also a severe lack of valid or reliable evidence. We certainly weren't there. And so these give us clues to prehistoric life. But of course, there's always some doubt to it because the evidence is lacking. Now, with that comes extinction, our classic extinct animal, the dinosaur. Well, extinction occurs if you're unable to evolve quickly enough. You haven't had the ability to change. Well, I guess the question is why? Why might you need to evolve? Why might you need to change? Well, a whole host of things. Perhaps there's some environmental change. It gets hotter, colder, the pH changes, the carbon dioxide levels change. Perhaps there's a new predator and he's very good at eating you. Perhaps there's some disease. Perhaps there's new competition, a new species perhaps. Perhaps there's some worldwide cataclysmic event, a meteorite. If any of these happen, and you're unable to keep up, you will go extinct. Your species will die out. Well, that leads on to our final point. What is a species? How does it come about? This is speciation. This is the development of new species. And a species is simply defined as an organism that can successfully mate and produce viable offspring. To put that another way, a human and a chimpanzee are different species. That's my chimpanzee very badly. They are different species because they cannot viably reproduce. They can't have an offspring which itself can reproduce. That might be because of physiological reasons, but it's normally genetic reasons too. Well, how do new species come around? Let's take box 1. We've got an island with some black dots. These black dots represent a species, and they're all exactly the same. They're in 2 different populations, but they're exactly the same species. Something happens. It could be a new mountain range. So it could be our environmental change. It could be perhaps a predator comes in between them. There's new species forming based upon this. How? Well, some kind of barrier exists. If you give that enough time, and the environment perhaps changes over that time. So, for example, here in the southern region, it's a bit rainier. In the northern region, it's a bit sunnier. If you give that enough time, those 2 species, which are the same, might begin to adapt slightly differently to their environments. If that adaptation occurs over enough time, perhaps they change so much that the differences become huge. And if that barrier were to go away and the 2 populations were allowed to remingle, they would have changed so much that they're no longer going to breed with one another. They have become a new species. So a population has some kind of barrier over enough time in different environments, we can get ourselves new species. Okay, we'll end on this. Exam question 5, life on Earth. We've got ourselves a large amount of evidence that evolution's taking place. But scientists are uncertain about how life started on Earth. For 2 marks, why? Well, we've got a lack of valid or reliable evidence, or soft-bodied, simple organisms are destroyed. They're not around. Now, for the second part, salamanders are these terrestrial amphibians, and the diagram shows the distribution of the 4 different species. So we got species Z, Y, X, and W. Originally, there was just one. Sorry, I've cut the picture off a little bit. There was just one species. How did all 4 come around? Well, our clue here is the mountain ranges. Presumably, there's been some type of isolation by these mountains. Perhaps they've formed over the last few millennia. The isolation then allows for genetic variation. Perhaps each place was slightly different, and different variations were more likely to be successful. And the driving force behind that? Natural selection. Perhaps nature selected certain genes in different populations to have a more advantage. That natural selection causes differences in each population, and so eventually if the barriers were taken away or the species were interact, interbreeding would no longer happen. So the isolation of the mountain range of this barrier allows for slightly different conditions. Natural selection with slight genetic variation causes different genes to be more prudent, to be more successful. They're passed on. Over time, differences occur. And if the barriers were to ever be lifted, the interbreeding would no longer happen. And for our last question, we've got ourselves a question on fossilization. How do fossils provide evidence that species alive today have evolved from a simpler organism? Well, first of all, fossils show our organisms that lived a long time ago. We see changes over time. Older organisms are simple. But the fossils do remain similar. So we can see with present day species some similarities, but the old ones do have significant changes, which essentially give you a timeline of how they've changed. Okay, we've got two pictures of gulls, two species. Both species are found in the UK, but two species cannot interbreed. Scientists believe that these two species evolved from a common ancestor. The map shows a view of the Earth from above the North Pole, and it shows where these two species are found. Now, I've put the picture just here. Suggest an explanation for the development of these different species. Again, if it was 6 marks, we're going through what we previously talked about. There's some type of isolation, a barrier. Perhaps it's, in this case, the sea. That then causes natural variations. You might put different alleles. Because of different environmental conditions, to be good or bad. Natural selection will then pick the most suited. It will act upon these, and so only these genes are passed on. If natural selection is picking different genes based on two different conditions, then over time, we're likely to find a new species forming. We are not allowing interbreeding. Okay, ladies and gentlemen, that is the last question for the second part of biology, and that concludes the biology apart from the night before the exam hangout. Again, we hope you found this really useful. The video is saved in the catch-up area, and I'll remain online for any further questions. Have a good evening.